from Laurent Lagrost. He is from INSERV, Université de Bourgogne.
Thank you. Good morning. It is my pleasure to be here today, and I would like to thank the organizing committee for this invitation. So, ladies and gentlemen, 65 to 70% of our body is made of water. Of course, water is essential for life, but in the meantime, water is a major challenge, at least at the molecular level. How can we handle hydrophobic lipophilic molecules in the aqueous phase? Our diet contains a large variety of hydrophobic molecules, hydrophobic nutrients. Some of them are actually essential nutrients, including, for instance, omega-3 fatty acids or liposoluble vitamins. So a key question here is to know how these hydrophobic compounds can be transported in the aqueous bloodstream. How can they reach the target tissues? How fatty acids can reach muscles, adipocytes? How vitamin E can go across the blood-brain barrier. In the opposite, some of the hydrophobic molecules are harmful molecules, at least when they are present in excess. Of course, it is the case for cholesterol. Cholesterol in excess has to be removed from the periphery. It has to be transported back to the liver through a unique pathway, the reverse cholesterol transport pathway. And under certain circumstances, for some reasons, gram-negative bacteria in our gut can translocate. They can go across the gut barrier. They are highly pro-inflammatory molecules at the surface of these gram-negative bacteria. We are not able to catabolize these lipopolysaccharide molecules. There is only one way to get rid of LPS, to bring them back to the liver and then to the gut lumen using a specific pathway, and I would like to discuss these points today. So, but first, from a physicochemical point of view, here are some of the structures that hydrophobic portions can actually form in water. These molecules are rarely found, at least only at trace amounts in water as free monomers, just because they are hydrophobic. They cannot be solubilized in water. They rather form more complex structures, including small micelles or bigger lamellar vesicles. They can also form more complex and more disordered structures, such as inverted aggregates or mixed aggregates. But back to the real world, back to the vertebrate species, and in particular back to the human situation. In our blood plasma, our lipids are transported mainly as part of the plasma lipoprotein particles. And we may consider that, in fact, these lipoprotein particles constitute the ultimate carrier of hydrophobic portions in vertebrate species. So here is one lipoprotein particle. It is a spherical structure. It has a highly hydrophobic core, which is actually made of neutral lipids, cholesterol esters, and triglycerides. This highly hydrophobic core is surrounded by a monolayer of amphipathic molecules, phospholipids, and esterified cholesterol. And also in the surface, you can see here the proteins. These are the apolipoprotein molecules. Apolipoproteins are very important to consider. First, they can stabilize the overall lipoprotein structure. They can also modulate, activate, or inhibit lipolytic enzymes or lipid transfer proteins which interact at the lipoprotein surface. And the last but not the least, they can be specifically recognized by cellular receptors. So we know this structure very well thanks to their ability to transport cholesterol. And each of us is well aware of low-density lipoprotein LDL as a bad guy and high-density lipoprotein HDL as a good guy in the prevention of atherosclerosis and cardiovascular diseases. What I would like to emphasize this morning is that in addition to lipids, cholesterol, triglycerides, these lipoprotein particles have the ability to bind and transfer a number of other amphipathic compounds, including liposoluble vitamins, vitamin E, vitamin A, lipophilic drugs, and also lipopolysaccharides or bacterial endotoxins. So here are two distinct lipoprotein particles. They are in the water environment of blood plasma. These lipoprotein particles can rapidly exchange lipid species, and it is actually a bidirectional exchange and transfer of lipid molecules. This type of rapid transfers can also occur between lipoproteins and cells. It cannot occur spontaneously, just because we are dealing here with highly hydrophobic molecules. It just cannot be transported as free monomers in water. But these transfers do occur, and they occur because of a specific family of genes coding for the lipid transfer lipopolysaccharide binding proteins. At least 3 members of this family are present in our plasma: phospholipid transfer protein, PLTP, the cholesterol ester transfer protein, CTP, and the lipopolysaccharide binding protein, LBP. And all these proteins share the same type of structure. They are boomerang-shaped proteins with specific properties. So here is PLTP, phospholipid transfer protein. Initially, it was discovered and described for its ability to bind and transfer phospholipid molecules between lipoprotein particles as well as between lipoprotein particles and cells. Today, and over the last years, we learn much more about PLTP. Today, we know that this protein has the ability to bind and transfer a number of other amphipathic compounds, including esterified cholesterol, vitamin E isomers, lipopolysaccharides, and so on. So first, as an example, I would like to briefly discuss the combined role and implications of lipoproteins and phospholipid transfer protein, PLTP, in vitamin E transport. We know a lot about vitamin E. It is a lipophilic antioxidant molecule. It is made of a mixture of distinct isomers, tocotrienols, tocopherol, with alpha-tocopherol being the most biologically active form of vitamin E. Here is the brain. We know that the brain contains high amounts of vitamin E, and we know that the brain has high vitamin E requirement. Brain contains elevated amounts of highly oxidizable polyunsaturated fatty acids. And I would like to show you a few data indicating that the brain actually relies, at least in part, on vitamin E lipoprotein complexes and PLTP for normal function. So these are earlier studies which were conducted in our group by Catherine Desrumeaux. It was conducted in collaboration with Professor Jiang and Professor Toll at the Columbia University in New York. We studied here genetically engineered mouse models. These are PLTP knocked out mice with complete PLTP deficiency, while normal wild-type mice have a naturally high PLTP activity. And as shown here on the right-hand side, no PLTP activity in the body of these animals means 30% decrease in vitamin E content of the brain. It is associated with accumulation of oxidized lipid derivative in the brain of the animals, here with accumulation of lipofuscin pigments in the brain of PLTP knocked out animals. Interestingly, it has consequences on behavior of these animals. It was assessed by using distinct maze paradigms. In collaboration with Professor Dessort in Nancy, we use the elevated plus maze for anxiety measurement. And more recently, we use the Y maze, which allows evaluation of the short-term spatial memory. And it was conducted with Dr. Desrumeaux in Montpellier. In all cases, we observe that the PLTP deficient trait, which is actually associated with partial vitamin E deficiency, is associated with changes in behavior of these animals. They are more anxious, spending more time here in the closed arms of the paradigm. And on the right-hand side, we used a pathomimetic model of Alzheimer's disease. In this case, animals were injected with an oligomeric preparation of beta-amyloid peptide, And we could observe that PLTP knocked out mice have a complete loss of short-term spatial memory as compared to wild-type mice or mice injected with a vehicle. So, okay, so it indicates that there is a complete pathway for tissue incorporation and transport distribution of vitamin E in our body. Here is dietary vitamin E. And first, vitamin E has to be incorporated into intestinal lipoprotein particles, which is a key step which can be, at least in part, facilitated by PLTP. Here are the lipoprotein particles carrying vitamin E from the gut to the liver. The liver is a major central organ in vitamin E metabolism, and thanks to alpha-TTP, alpha-tocopherol transfer protein, PLTP, this protein have the ability to incorporate vitamin E in liver VLDL particles, and then here are these particles with vitamin E, and in the end, vitamin E has to go across the blood-brain barrier to reach the brain. So as you can see here, it is a multi-step process, and each of these steps actually involves PLTP. So it's not only a matter of amounts of vitamin E which is brought in the diet, it is also a matter of an optimal pathway through which vitamin E at the right amount, at the right level, can be brought to the right tissue and at the right time. So now I would like to discuss briefly the role of PLTP and lipoproteins in lipopolysaccharide detoxification. So here is 1 gram-negative bacterium. It contains a lipopolysaccharide molecule in the outer wall. And there are huge amounts of bacteria and lipopolysaccharides in our gut. There are more gram-negative bacteria in our gut than we have human cells in our body. 1 bacterium, these are 100,000 molecules of LPS, so a huge amount of LPS, which is present in the gut lumen. Unfortunately, we cannot catabolize these LPS molecules. And when they are present in our bloodstream, they can activate human cells through interaction with the TLR4 receptor, which is located at the surface of the immune cells. And activation of TLR4 triggers inflammatory response, which at least under certain circumstances can be or may be harmful. So we cannot catabolize LPS, but we can transfer LPS from the outer wall of gram-negative bacteria to circulating lipoprotein particles. It is a highly efficient pathway, and again, it is mediated by 1 single factor, the phospholipid transfer protein, PLTP. What about the importance and the relevance of this pathway, which is named reverse LPS transport pathway in our group today? Is it highly efficient or not? Yes, it is. And these are very recent studies recently published. They were conducted in collaboration with Professor Brunot and Professor Denard here at the University of Burgundy. We could achieve a dual labeling of lipopolysaccharides. It was performed by using a bimodal molecular probe, which was specifically designed for both fluorescence and nuclear imaging. So this labeled LPS were injected to mice through the jugular vein. And here is a picture we obtained a few hours following the injection in the jugular vein. As you can see, the bulk of LPS was actually removed from the periphery, and it was transferred towards the liver, which means that these animals have the ability to disaggregate LPS in the periphery and to bring it back very quickly to the liver. Here are a few key steps of this process. This is the association of lipopolysaccharides with HDL. Again, we are back here to the mouse model, wild-type mice with high PLTP activity, and PLTP knocked-out mice with no PLTP activity. And as shown here, when PLTP is expressed, it can markedly accelerate the association of lipopolysaccharides with HDL, and it's much slower. in the PLTP knocked out mice. It has a direct impact on the overall kinetics of LPS in plasma. Clearance of LPS is significantly slower in PLTP knocked out mice as compared to wild-type mice, and it has a direct consequence on inflammation. More LPS at one given time means more inflammation, and indeed we could measure significantly lower levels of IL-6, interleukin-6, in wild-type mice as compared to PLTP knocked out mice. And here is the last step of the process, biliary excretion of LPS. So here we used a brand new assay, which is named EndoQuant, which was set up in our laboratory. It is a quantitative and sensitive assay to measure LPS concentration using LC-MS analysis. And as shown here, the amount of LPS excreted in the bile of wild-type mice is much higher as compared to PLTP knocked out mice. Those PLTP expression means that one can get rid of much quicker of LPS. So here, back here to a general scheme. So this is the gut lumen with gram-negative bacteria under certain circumstances, including, for instance, bowel diseases or including changes in the diet, for instance. These bacteria may translocate, may go across the gut barrier either as an old bacteria or as smaller LPS aggregates. So once in the circulation, these LPS are highly pro-inflammatory, again activating TLR4 in immune cells. So LPS has to be quickly disaggregated, and PLTP is involved. They have to be transferred towards lipoprotein particles, then producing a double effect, first a dual effect, first neutralization and second transport. And finally, this LPS can be detoxified in the liver through biliary excretion. So PLTP has the ability, again here, to play a key role transferring, shuttling LPS molecules between distinct entities. And we are quite convinced today that by using lipoproteins associated with PLTP, we should be able to enhance this reverse LPS transport pathway, thus reducing the inflammatory response. Actually, in collaborative studies with a French company, the Bioprotein Technologies company, close to Paris, we were able to produce large amounts of fully active recombinant human PLTP. This PLTP can be obtained as a purified protein, and it was used as a therapeutic tool in animal models. Models of sepsis, mouse model of sepsis, were injected with PLTP, and we observed two main things. First, there was a major decrease in the inflammatory response of these animals, and second, we observed a major increase in survival of these mice. So as conclusions, we are quite convinced today that functions of lipoproteins and lipid transfer protein extend beyond, and maybe I should say far beyond, cholesterol transport and atherogenesis. The second point is that fortunately, we know a lot about lipoprotein metabolism. It has been, I should say, nearly deciphered, and thanks to the role of lipoprotein metabolism in cholesterol transport, they have been extensively studied in the context of atherogenesis and cardiovascular diseases. Fortunately, also today we know and we hold effective lipid-lowering drugs, statins for instance, and we know a lot about dietary manipulations which are susceptible to modulate, modify the profile, the quantity, the kinetics of lipoprotein particles. So now, what we want to explore is the role of lipoproteins as a field, not only atherosclerosis and cardiovascular diseases, and we do believe today that lipoproteins may bring new insights and solutions into the prevention and treatment of endotoxemia, sepsis, bowel diseases, metabolic diseases, and neurodegenerative disorders. At least these questions are currently addressed through our Lipstick LabX. Lipstick LabX is part of the Investment for the Future program of the French government. Lipstick means lipoproteins and health, prevention and treatment of inflammatory diseases and cancer. And it is a program involving 4 distinct universities in France with 23 distinct groups throughout our country. And we have tight and long-lasting collaboration with Vitagora. There are 5 main axes, and I will end with that. First axis, role of lipoproteins in the absorption, transport, and distribution of natural bioactive molecules. I said a little bit today on vitamin E, but we have some interest for other natural bioactive molecules. The second point is dealing with lipids and lipoproteins in the initiation and progression of cancer and inflammatory diseases. We know a lot about HDL as, again, the good guy, LDL as a bad guy in atherosclerosis. But one of us recently in the LabX has contributed to the demonstration that HDL cholesterol actually correlates negatively with the incidence of colorectal cancer. So quite a new area which deserves further attention. Third point, lipoprotein-mediated drug delivery, in particular in the treatment of inflammatory diseases and cancer. It is a very exciting area. We know today that some of the anti-cancer drugs which are used to treat patients are lipophilic molecules. For instance, paclitaxel is a well-known anti-cancer agent, and we know that it is actually located inside the hydrophobic core of lipoprotein particles, which means that if we are able to modulate lipoprotein metabolism, lipoprotein uptake by the cells, in particular cancer cells, we may help to reduce toxicity of the treatment and increase its efficacy. Fourth point, I said a lot about that today, endotoxins and lipoproteins, neutralization and elimination. And our main goal here, of course, is to turn down the inflammatory response. And finally, through this LabX, we set up collaborative studies with our colleagues from economy and from human sciences. And we are currently searching for medico-economic evaluation and transfer of knowledge. Finally, my special thanks go to my colleagues who took part in the studies I presented today, and also my colleagues who are currently involved in our group in this long quest, and I thank you for your attention.
Thank you very much. We have time for one short question. Luc?
Thank you, Laurent. I do have many questions, but just one. In your PLTP KO mice, you showed that there is a partial deficiency in the vitamin E content in the brain. So does that mean that there is other system to transport vitamin E from the gut to the brain?
Of course, there are. I mentioned alpha-tocopherol transfer protein, which is well known in the field. Alpha-tocopherol transfer protein has the ability to incorporate vitamin E in lipoprotein particles, as PLTP is doing, and it can also probably do something in the brain. And we do believe it is the same with PLTP. It's not only a matter of quantity of uptake of vitamin E by the brain. It is also a matter of distribution, and very often subtle distribution of vitamin E inside the cells. For instance, it depends whether it is located in one membrane domain or the other. So it's not only a matter of decrease in quantity, it's also a matter of, I would say, quality and distribution of vitamin E. Alpha-tocopherol? What do you mean?
Since the first step is to go from the gut to the gut-brain barrier, do you have also alpha-tocopherol in the gut?
Yeah, of course, for sure you need alpha-tocopherol in the gut to bring it to the gut and to be transferred in subsequent steps. Yeah,
okay.
Okay, thank you very much. So.